Biological specimen can range from several millimeter to several nanometers. Using light microscopy, we can visualize specimen up till 200 nanometer or 2.2 micron. If a specimen is smaller than that, then the specimen and the resolution limit is crossed. Everything is lost in the diffraction blur. We cannot see those specimens or resolve these specimens using light microscopy. We need electron microscopy for that. Electron microscopy comes in two flavors: transmission electron microscopy and scanning electron microscopy. Both uses electron but in a different way, and both are equally important for biological application. In this video we are going to talk about both these microscopic techniques, so stay tuned till the end and enjoy. SEM is an important microscopy technique that is used to obtain high resolution images and detailed surface topology information of a sample. Now, it's a type of microscopy that uses focused beam of electron instead of light to scan the surface of a specimen. It could be a biological as well as non-biological sample as well. What is the resolving power of SEM? So the resolution power of SEM is quite high, and it is in the range of less than a nanometer to several nanometers. Remember, the resolution limit of a light microscope is about 200 nanometer classically. Why SEM has a better resolving power than a normal light microscope? That is something we have to understand. The light microscope limit of resolution D is provided as 0.6 lambda by Na or n sin alpha. So this is the resolution formula. Now let's say there is a microscope which has which is operating at a objective of 60x, so roughly the Na would be 1.2 to 1.4. Here we are just putting 1.2. So roughly the limit of resolution at lambda 400 would be 200 nanometer. That simply means if 2 points are 200 nanometer apart, one can resolve that using this microscope. If they are closer than this distance, nothing can be resolved. But in case of SEM, the lambda of electron is much lesser, and this is a function of the accelerating voltage that we provide. Greater the voltage, lesser is the wavelength. Now if we can just get a quick idea about the lambda after putting all the values that we shown here and putting V equal to one particular EV, we can see the lambda is about 1.2 nanometer. So you can understand if the lambda is smaller, the resolution would be much better, right? So this is why the scanning electron microscope has much better resolution compared to a light microscope. So now let's look at the optical components. So this is how it look like, pretty much like a tube, inverted tube, and it's basically a vacuum chamber where the entire microscope lens, electron gun and etc. are placed. So first you would see the electron gun, which is the source of the electrons. For SEM and the TEM, similar kinds of electron guns are provided. If you want more details about electron gun, watch the video on TEM. So it would generate the electron beam that would go through the several lens array. So there are a couple of condenser lenses here. The first condenser lens, the spray aperture, eventually the second condenser lens. Eventually they would pass through a deflection coil. This deflection coil help in the raster, or means like scanning the surface topology of a specimen. Here are the objective lenses in yellow. All of these lenses are actually electromagnetic lenses. If somebody want to accelerate electron, they have to use the electromagnetic lenses, right? That is how the electron beam could be focused on a narrow volume on the specimen surface, and it can be rastered on a specimen surface in a desired area. This is the sample. It's mounted on a particular chamber which can be tilted into different rotation to get the optimal plane. And these are something called backscatter electron detector, these are secondary electron detector, and these are X-ray detector. So these are three types of electron that are generated when the electron beam is basically interacting with the sample, and all of these has information regarding the specimen topology. So three most important component is first the electron gun, which produce the electrons, then the electromagnetic lens array, which has the condenser and the objective lens, which is useful for focusing the beam. Third is the scanning coils, which move the beam over the sample, and the fourth is the detector that captured the emitted electrons or emitted signals and creates the image. So if we really summarize the working principle of SEM, the three important component was electron beam generator or electron beam, then basically the scanning modality, which is done by the electromagnetic coils and the objective lenses, and last thing is the interaction with the specimen, which can be detected by several kind of detectors, secondary electron detectors and backscattered electron detectors, etc. Now, depending upon the voltage of the electron, so the acceleration voltage of the electron, the volume of image and the penetration depth would vary. So here you can see if the accelerating voltage of the electron is 15 keV, then the penetration depth is about 6 micron, whereas it is becoming 0.5 micron if the accelerating voltage becomes 5 keV. So lesser the accelerating voltage, lesser is the penetration. So simply we can understand higher voltage increases the penetration depth but reduces surface resolution. Lower voltage improves the surface details and it gives a shallower penetration. So both are true, but the thing is it really matters what we are trying to image, and based on that we have to apply the acceleration voltage in this particular microscope. Now let's talk about the type of electrons that are generated. First type of electron is secondary electrons. That is basically generated when the electron beam is literally kicking out electrons from the outer orbit of particular sample. So these secondary electrons could be detected by secondary electron detectors. Then there are backscattered electrons. So these backscattered electrons are those electrons which penetrate an atom but eventually gets elastic scattering and eventually move in the backward direction. So these are backscattered electron. They offer information about composition, and they have other by changing or collecting the backscattered electrons, we can literally have more contrast in that image. Also there are X-rays that are used for elemental analysis with energy dispersive X-ray spectroscopy, but this is beyond the beyond the scope of this particular small video. Now every sample that has to be put under the SEM, every biological sample, because we are biologists, right, has to be coated, because mostly biological samples are non-conductive. That's why it has to be coated with, let's say, osmium tetroxide, gold, platinum, or even carbon thin layer of that. So sputter coating equipment like this is used where a coating happens in a vacuum chamber. Now other than that, there are special fixation and dehydration paradigm that has to be done. Special fixative agents such as Buin's fixative agent has to be used, for example, in case of SEM. There are certain advantages and disadvantages. Obviously SEM is best if you want to look at the surface topology, but you want to look at inner cellular details, organelles, etc. SEM is not the key. In that case, TEM is the key. It can analyze a wide range of materials, ranging from biological materials to metals to polymers, etc. So that is why it has a versatile usage in the field of nanomaterials, biology, and also condensed matter physics. But it has several disadvantages. First is it cannot image a viable cell. It can only image a cell after it is fixed, it has died, and it is processed in a tons and tons of way. Okay. Another thing is like only surface details are visible, not suitable for investigating fine changes in the internal structures. Again, it requires a costly and fancy equipment with a lot of like technical skills. So these are kind of like a downside of these equipments. But anyway, be it biology or be it other disciplines, SEM is a wonderful instrument to look at surface topology. So I hope this video was useful. If you like this video, give it a quick thumbs up. Electron microscopy is a technique which uses a high energy electron beam which is transmitted through a specimen to form an image. TEM is providing incredibly high resolution, allowing the observation of finer details within a cell. It can literally provide a magnification up to 10 million times. That's amazing, right? That is why finer details inside the cell, like ultrastructure of organelles, can be determined using this transmission electron microscopy technique. Literally, cell biology is blooming due to this particular technique itself. So if this technique was not there in history, cell biology wouldn't have been advanced to this level. Now, typical light microscope offers a resolution of 200 nanometers, whereas a transmission electron microscope offers a resolution which is 0.1 to 0.5 nanometer, several thousand times more compared to a light microscope. In this video, we would try to understand how does TEM can offer this unprecedented resolution. Now let us quickly go through the advantage and disadvantage of the microscope. So basically it offers high resolution, it has high magnification, and it has versatile imaging applications. It can image nanoparticles, crystals, also biological specimens. But limitations are plenty. Sample preparation is cumbersome. Any biological specimen has to be as thin as 100 nanometer such that electron beam can pass through. Vacuum chamber has to be created because electron beam other would scatter. Radiation damage is always a hazardous risk in this kind of technique. Complexity and cost-wise, it's not really economic. Now let's talk about the sample preparation for a transmission electron microscope. So first, ultra-thin sectioning is done in a ultra microtome, which has a microscope attached to it. So you literally section the samples as thin as 100 nanometer in thickness. After that, they are collected on a water bath and then mounted on a copper grid as shown here. This copper grid is then placed on a cartridge which is placed further into a sample holder. The sample holder can be rotated in 3 directions to fit the sample and orient the sample in the path of the beam. Now let's look at the beam path to understand the technique much more better. So here is the overall envelope of a electron microscope or the transmission electron microscope. So here is the electron gun, which is the source of the electron. Here are the condenser lenses. Here are the condenser apertures. This is the objective lens. This is the sample holder. This is the intermediate and projector lenses. And lastly, it gets projected into the fluorescence screen. Now, electron gun generates the electron beam which pass through the condenser, get focused onto a narrow region in the specimen. It passes through the objective aperture and the objective lens, get magnified, and eventually through the intermediate lenses and projector lenses get projected onto the fluorescence screen. So this is the overall light path or electron beam path actually in this case of the transmission electron microscope. Now, one important thing to note that the entire array is placed inside a vacuum chamber. Because if it is not in a vacuum chamber, then electron would collide with different components and would create huge scattering. Now, all these lenses which are used to accelerate the electron beam are actually electromagnetic lenses, because that's how you can literally focus or move electrons in a particular vacuum field, right? Now let's talk about the components of the electron microscope. So there are basically high voltage sources, vacuum systems, microscope columns, detectors, and particular softwares. So here is the electron gun, here is the condenser lens, here is the objective aperture, sample specimen port, here is basically the objective lens, here is the diffraction lens, here is the intermediate lens, here is the projector lens, here are the binoculars, and here are the fluorescence screen where the image is formed. Underneath that, one can also have charge coupled device as an electron imaging system. So it is basically an electron detector. Now you can see the electron beam falling, passing through the specimen, and ultimately going and hitting the fluorescence screen. It's important to note that there is one concept of accelerating voltage. Accelerating voltage is a voltage or a field potential which is applied to accelerate the electron through this narrow tube. Greater the accelerating voltage, smaller would be the electron wavelength, and improved would be the resolution. So basically, higher voltage would be better resolution. Now let's look at the electron gun. Electron gun is an element which has tungsten wire. So electron gun produces the electron beam. So it can have different material like tungsten filament, field emission gun, or even lanthanum hexaboride crystal. Ultimately, electrons are produced, and the important point is the gun is connected to very high voltage. And now we understand what is accelerating voltage. Now you can see it falls onto the condenser aperture. Through the aperture, it falls onto the specimen holder. So the job of these yellow condenser lenses is to focus the beam to the sample, right? So condenser lens ensures that the beam is narrowly focused to the proper orientation of the sample. You can also use the sample holder and twist it, turn it into different angles to orient your sample in a proper situation. Now the condenser lenses are basically nothing but electromagnets. So now we are going to talk about the other components. So here you can see through the specimen the electron beam is passing through. It's transmitted, right? Because it is transmission electron microscopy. Now it is passing through the objective aperture, and ultimately basically it is moving through this particular objective aperture and moving to the objective lens. Here is a higher magnification view of your specimen port, which can be rotated. Here is the cartridge, and you can see on top of the cartridge in a top view you can see the grid, and a clamp is fixing the copper grid into its location. Okay, now we can see there are a bunch of lenses which actually help in the projection of the electron beam to the fluorescent screen. There were objective lens, diffraction lens, intermediate lens, and projector lens. All of these lenses are important to focus this beam to the fluorescent screen or the detector. So the image of the specimen with its assorted part appears on the screen based on the density. So if we look at the overall electron micrograph of a mitochondria here, you can see some regions are dark, some regions are light. The regions which are dark has very high electron density. The regions which are light has low electron density. Now there are different electromagnetic lenses. We talked about condenser lens, objective lens, and the projector lens. These three are really important. Condenser lens focus the beam onto a narrow region. It ensures the electron beam is finely tuned before it hits the specimen. So typically 2 or 3 condenser lenses are there. Objective lenses are also important because the beam pass through that, basically pass through the objective lens and get magnified. So this is the first stage where the magnification happens. Then the lens, this particular objective lens largely determines the resolution of the TEM image. Then there are projector lenses that directs the magnified image into the viewing screen or the CCD, and it helps to achieve additional magnification. So projector lens also magnify the image further. Now let's talk about the physics a little bit to understand the resolution. So any case, the resolution of a microscope is given as d equal to 0.6 lambda by n sin alpha, where n equal to refractive index, alpha is the aperture angle, lambda is the wavelength. Now if you do the math and use a wavelength of 400 nanometers and use an objective whose refractive whose basically numerical aperture is 1.2, in that case the resolution is 200 nanometer. So a light microscope simply can tell apart 2 points which are 200 nanometer apart. But what about TEM? TEM can improvise this capability to several thousand folds. Why? Because TEM uses electron. Electron has much lower wavelength. So wavelength of an electron is calculated like lambda equal to h by 2 MeV. So here if we plug in all the values that are provided here, we would try to see the wavelength. It is 1.227 nanometer, which is several hundred nanometer lower than a particular visible light. Now if you use a higher accelerating voltage, the lambda of electron would be even lower. So resolution would be even higher. So lower the lambda, smaller the small distance that could be resolved apart. So better would be the resolution. This is how TEM is much better in terms of resolution. But the big disadvantage of TEM that we cannot capture living dynamics inside a cell. So we cannot literally look at a mitochondrial fission fusion event using an electron microscope because the specimen has to be killed. Imagine a live imaging setup where you are tracking a movement of a cell. You cannot do that with electron microscope, even if the resolution is better, because the specimen has to be killed. So despite of all the advantages, there are limitations of these TEM instruments, and we need to know the limitations being a researcher. There are different applications of TEM beyond biology. So obviously in biology, looking at the architecture of the cell, viruses, proteins, etc. could be useful. In terms of material sciences, in order to study crystal defects of the nanomaterials, this particular technique is very useful. Nanotechnology and physics also has a lot of applications of transmission electron microscopy. So beyond biology, transmission electron microscopy is also really useful. So I hope this video was useful. If you like this video, give it a quick thumbs up. Don't forget to like, share and subscribe. You can find our notes and in flashcards in Instagram or Facebook page. Links are provided in the description. You can click on the super thanks to help us our channel grow. Your small contribution is our big motivation. See you in next video.